Have you ever looked up at the night sky, lost in the sheer, overwhelming vastness of it all? That feeling of smallness, of wonder, it's a little like looking at DNA. It's beautiful, isn't it? A mesmerizing complexity hidden within each of us, a secret world operating within every single cell. It's something we take for granted, yet it's arguably the most important molecule on Earth. For decades, its true form remained a mystery, only revealed about 60 years ago. Imagine taking the DNA from just one of your cells, stretching it out, untangling it. It would be taller than you are. Now pause for a moment. Consider this. You have roughly 50 trillion cells in your body right now. If you linked all the DNA within those cells end to end, it wouldn't just reach the sun. It would stretch to the sun and back, over 600 times. Let that sink in. We're talking about deoxyribonucleic acid, DNA, the blueprint of life. It's the instruction manual, the complete and utterly detailed code, containing 6 billion letters that dictate everything you are. Not just your physical traits, but your predispositions, your vulnerabilities, the very essence of your being. And it's not unique to you. This same code, with slight variations, governs the lives of nearly every other living thing on this planet. If you're human, then every single somatic cell, that's every cell that isn't a sperm or egg cell, contains 46 chromosomes. Each chromosome is a tightly wound package, a beautifully organized collection of one incredibly long DNA molecule. These chromosomes reside within the nucleus, the control center of the cell. Now, DNA isn't alone in this world of molecular biology. It has a cousin, ribonucleic acid, RNA, and we'll be exploring its role soon enough. But first, let's take a step back. Remember those fundamental building blocks of life: carbohydrates, lipids, and proteins. Those were essential, absolutely. But nucleic acids, DNA and RNA, they operate on a different level. They have the most complex job of all. They are at their core polymers. That means they're built from many small, repeating units. In the case of DNA, those units are called nucleotides. Link enough nucleotides together, and you have a polynucleotide chain. Before we start assembling this microscopic masterpiece, let's break down what makes up a single nucleotide. 3 key components: a 5-carbon sugar molecule, a phosphate group, and one of 4 nitrogenous bases. The deoxyribo part of DNA's name comes from that sugar, deoxyribose. But the real magic, the genetic code that defines you, resides within those 4 nitrogenous bases: adenine, thymine, cytosine, and guanine. These aren't just random letters. They are the alphabet of life. However, DNA doesn't exist as a single, solitary strand. It exists as a pair, 2 polynucleotide chains intertwined, linked together in a spectacular double helix. Think of a spiral staircase, elegant and strong, but on a microscopic scale. It's a structure that has captivated scientists for generations. And like any great structure, it needs a strong support system. That's where the sugar and phosphate groups come in. They form the backbone of the helix, running along each side but with a crucial twist. The 2 strands run in opposite directions. Imagine 2 roads heading in opposite directions. That's what's happening here. One strand starts with a phosphate group attached to the sugar molecule's 5th carbon, ending with a free end at the sugar's 3rd carbon. This orientation is referred to as 5' and 3', a shorthand chemists use to describe the direction of the chain. The other strand is flipped, starting at the third carbon and ending at the fifth. It runs in the 3' to 5' direction. Always remember 5' to 3'. It's a little detail, but it's fundamental to understanding how DNA works. Now let's build our own double helix. The two strands are connected by those nitrogenous bases, forming what are called base pairs. But there's a strict pairing rule. Adenine always pairs with thymine, and guanine always pairs with cytosine. It's a perfect match, a lock and key relationship. Guanine-cytosine pairings are stronger, with 3 hydrogen bonds holding them together, while adenine-thymine pairings have only 2. The order of these base pairs, AGG to CCATG, is what makes each of us unique. Change the order and you change the code. To illustrate the scale, consider this. Chromosome 1, the largest of all our chromosomes, contains 247 million base pairs. If you printed out the letters of chromosome 1, it would fill a book with approximately 200,000 pages. And each of your cells has 46 of these DNA molecules. Put them all together, and you're looking at roughly 6 billion base pairs within every single cell. That's a library of information beyond comprehension. Quick test. Let's see if you're following along. I'll give you one strand of DNA, and you tell me the complementary strand. Ready? Here it is, 5, AGG to CCG3. Time's up. The answer is 3, TCCAGGC5. See? It's not so complicated. Because each base has only one partner, you can predict the complementary sequence with ease. But can we use RNA to create the same sequence? Not quite. RNA is similar to DNA. It has a sugar phosphate backbone and nucleotide bases, but there are key differences. First, RNA is typically single-stranded, no double helix here. Second, the sugar in RNA is ribose, which has an extra oxygen atom compared to deoxyribose. That's why it's called ribonucleic acid. And finally, RNA doesn't contain thymine. Instead, it uses uracil, which pairs with adenine. RNA plays a vital role in protein production and DNA replication, but we'll dive deeper into that later. Now, let's talk about the people who unraveled this incredible mystery. It wasn't a single eureka moment, but a century-long journey involving numerous brilliant minds from around the world. You often hear the names James Watson and Francis Crick, but that's only part of the story. They didn't discover DNA, nor did they discover that DNA contained genetic information. That honor belongs to Friedrich Miesser, a Swiss biologist who, in 1869, isolated a strange gray substance from the nuclei of white blood cells. He obtained those cells, disturbingly, from used bandages collected from a hospital. For science, he persevered. After removing the lipids and digesting the proteins, he was left with this unknown substance, which he called nuclein, later known as nucleic acid. He didn't know its function or even its structure. Fast forward nearly a century, and enter Rosalind Franklin, a young biophysicist in London. Using a technique called X-ray diffraction, Franklin produced stunning images of DNA and may have been the first to confirm its helical structure. She even determined that the sugar phosphate backbone was on the outside of the molecule. So why is Rosalind Franklin a household name? 2 reasons. Firstly, she readily shared her data with her rivals, Watson and Crick. She even informed them that a triple helix structure was impossible, indirectly suggesting a double helix. Secondly, her crucial X-ray diffraction images were shown to Watson and Crick without her knowledge. Her work was published in Nature, but only after Watson and Crick had already published their papers, with minimal acknowledgement of her contribution. Tragically, Franklin died of ovarian cancer at the age of 37. It's believed that her exposure to radiation during her X-ray diffraction work may have contributed to her illness. The Nobel Prize cannot be awarded posthumously, and many believe she would have shared the 1962 prize with Watson and Crick had she been alive. Now that we understand the structure of DNA, let's explore how it copies itself. Cells are constantly dividing, and each new cell needs a complete and accurate copy of the DNA. This process is called replication, and our cells are remarkably efficient at it. They can replicate the entire human genome, that's trillions of base pairs, in just a few hours. First, an enzyme called helicase unwinds the double helix, breaking those hydrogen bonds between the base pairs, creating what's known as the replication fork. This opens up the DNA, exposing the individual strands to be used as templates. One strand is called the leading strand, the good guy strand, and the other is the lagging strand. The leading strand is easy. An enzyme called DNA polymerase simply adds matching nucleotides to the template strand, building the new strand continuously. But before it can start, it needs a primer, a short sequence of RNA. Nucleotides provided by another enzyme, RNA primase. The lagging strand is trickier. Because it runs in the opposite direction, DNA polymerase can only work backwards, creating short fragments called Okazaki fragments. RNA primase lays down primers, and DNA polymerase builds those fragments. Then, another type of DNA polymerase replaces the RNA primers with DNA. And finally, an enzyme called DNA ligase joins the fragments together, creating a continuous strand. DNA replication isn't perfect. There's about one error for every 10 billion nucleotides. But don't worry, our cells have a built-in proofreading system. DNA polymerase can detect and correct mismatched base pairs, ensuring incredible accuracy. Considering the sheer volume of DNA within each cell, miles and miles of it, it's astonishing that more mistakes don't occur. That's why scientists rightfully call DNA the most celebrated molecule of all time. So take a moment to appreciate this incredible molecule, this intricate code that defines you. Review this episode, let the complexity sink in. And next week, we'll explore how those 6 feet of DNA actually make you. Thank you to the entire Crash Course team for helping to make this episode possible. You can click on the links in the description to revisit any section of the video if you need a refresher. And please, share your questions and thoughts in the comments on Facebook or on Twitter.